Good morning. I'd like to begin by sharing a quote with you this morning. It's by Charles Sherrington. He says, "To recapture now and then childhood wonders is to secure a driving force for occasional grown up thoughts." And I feel like that quote, it just describes how I try and go about my experience in academia. So, last time we talked, we discussed neuronal communication and that neurons communicate electrochemically. And we talked about the electrical portion last time. We talked about the nerve impulse or the action potential. And what we're going to talk about this week is the chemical portion of neuronal communication. So it used to be thought that neurons actually grew into one another or made contact with one another. But there actually exists a gap in between the pre and post synaptic neuron. So the terminal buttons or bulbs of the presynaptic neuron and the dendrites of the postsynaptic or receiving neuron. And neurons communicate by transmitting chemicals at these gaps or junctions, which are called the synapse. Charles Sherrington was the one who coined that term to describe that specialized gap between neurons. And Sherrington's research was primarily studying reflexes. So we have an automatic muscular response to stimuli, right? A doctor takes that thing and taps on your knee area and your leg kicks out. So there are, sensory neurons that are going to receive some type of stimulation from the environment, and they are going to send their messages into the spinal cord. They're going to make contact with intrinsic neurons, which is going to send some information up until the cortex. But within the spinal cord, that neuron is also going to make contact with a motor neuron that's going to send its information to a muscle. Okay? And that's going to cause the behavior. So during Sherrington's research on reflexes, he discovered 3 important points. 1, reflexes are slower than conduction along the axon. Several weak stimuli presented at slightly different times or slightly different locations produce a stronger reflex than a single stimulus does. And as one set of muscles relaxes, another set becomes excited. So again, Sherrington observed a difference in the speed of conduction in a reflex arc from previously measured action potentials. He believed the difference must be accounted for by the time it took for communication between neurons. So what's happening at the synapse is what is causing the delay in time or the lag. So this was evidence that validated this idea of a synapse. Right? So the speed of conduction along an axon is about 40 milliseconds, but the speed of the conduction through a reflex arc is slower and more variable. Sometimes 15 milliseconds or less. And again, presumably the delay is what is happening at the level of the synapse. Okay, so here is synapse. Sometimes you'll hear it referred to as the synaptic cleft or a synaptic gap or a junction. Okay, so let's go to the second point here. So again, Sherrington observed that repeated stimuli over a short period of time produced a stronger response. Stimuli in rapid succession at the same point, right, or pinching the Achilles tendon of an animal multiple times would produce a cumulative effect in a stronger reflex. Sherrington also noticed that several stimuli, small stimuli on a similar location produced a reflex when a single stimulus did not. So this is spatial summation. So synaptic input from several locations can have a cumulative effect and trigger a nerve impulse. So we've been discussing neuronal communication from one neuron to a single neuron, but they exist in networks. A single neuron could be receiving input from hundreds or maybe even thousands of other neurons. So, so pinching the Achilles tendon of the dog that Sherrington would put in the harness to do his research at similar but not the same location, that could trigger an action potential, whereas touching a single area would not. So the takeaway here is that there's a summation process that, again, the voltage within a neuron needs to reach a threshold. So it needs to receive enough excitatory information for an action potential to be generated. The way to build up this excitation could be through rapid summation or summation of 3 different spots, but are all communicating with the postsynaptic neurons. So here's just an example, again, with a one-to-one connection, right? So here are dendrites that are receiving several impulses in quick succession. That'll change the voltage inside the cell enough for an action potential to happen and send its message to the next neuron. Or spatial summation. We have some information coming from this dendrite and this dendrite and this dendrite over here, right? So if there's going to be an excitatory message, it's opening sodium channels, right? It's changing the voltage within the cell. It's depolarizing that neuron in order for it to send an action potential. Right? Here's your axon. Here are your terminal buttons. Here's the receiving postsynaptic neuron, and there's a little gap here, which is the synaptic cleft. Another figure to show the buildup of excitatory information, or the change in the voltage within the receiving cell, right? So a single stimulus may slightly depolarize a neuron, right, or change the voltage, moving it towards its threshold, but it's not enough to cause an action potential. Remember, neurons, a lot of the neurons work as all or none. They either fire or they don't. They either reach the threshold and rapidly depolarize, or they don't, right? So here we see a single stimulus causes a little bit of a change. However, temporal summation, so rapid succession, 2 EPSPs will depolarize it more, but it takes a few stimuli, a few touches in rapid succession to reach the threshold. Again, once it does, rapid depolarization of the cell, right? Here are the sodium gates that are opening, and the nerve impulse or the action potential is traveling down the axon. Okay. Here's our refractory period. There could be some inhibitory messages that depolarize the neuron. It's moving back to its resting potential. So remember, EPSPs are excitatory postsynaptic potential, okay? It's excitatory, it's slightly depolarizing the neuron. It's not enough to reach the threshold, but several stimulation can change the voltage enough for a message to be sent. It's a very similar happening with spatial summation. Again, it's several weak stimuli together producing voltage change enough to reach threshold. Okay, so here's just another figure to help you visualize what is happening here, to make sure we have these labels down solidly. The presynaptic neuron is the neuron that delivers the synaptic transmission. So it's the neuron that's firing, right? The nerve impulse is moving down this axon. The postsynaptic neuron is the neuron that receives the message, and the communication happens between these terminal buttons at the end of the presynaptic neuron's axon and the dendrites of the postsynaptic neuron. Okay. Here is a blown up view of the presynaptic axon and its terminal buttons. And here you see this specialized gap called the synapse. Okay. Here is a receiving area of a dendrite on a postsynaptic neuron. So it's blowing up this spot right here. Okay. So, graded potential is a partial depolarization. Again, excitatory means more likely to fire. Hyperpolarization is inhibitory, it's less likely to fire. So excitatory and inhibitory postsynaptic potentials are graded potentials. They don't last very long, they can degrade. There needs to be a rapid buildup of excitatory potential to reach the threshold. Right, it's the cumulative effect of the EPSPs that are this basis for temporal and spatial summation. Now, a single neuron can receive both excitatory and inhibitory messages. It is the culmination, the summation of those messages that inform whether the neuron will send a nerve impulse. Just has to reach that threshold. Okay? Excitatory postsynaptic potentials open sodium channels, whereas inhibitory postsynaptic potentials either open potassium channels and allow some positive charge to leave the cell, or they allow chloride, which has a negative charge, to enter the cell. Okay, another thing that Sherrington noticed with research on reflexes in his dogs, that the foot of the dog was pinched, retracted, while the other 3 feet were extended. And so each foot of this dog, right, has both the flexor and extensor muscle. So here, while the flexor muscle is excited, the dog lifts up the leg, the extensor muscle is inhibited. As these extensor muscles are excited, the flexor muscles of these back legs are inhibited, right? So again, through these different networks, there are both excitatory and inhibitory postsynaptic potentials. And inhibitory information was at one point in time debated, but we now understand the importance of muscles being able to receive both of these types of messages. Okay. So again, an inhibitory postsynaptic potential is the temporary hyperpolarization of the postsynaptic membrane. Okay, so again, the chemicals that are being released at the synapse can carry messages that open potassium gates or chloride gates. So again, potassium leaves the cell of the postsynaptic cell and takes away positive charge. So the cell becomes more negative, becomes more polarized, it becomes less likely to fire. Or chloride, which has a negative charge, enters the cell and the outcome is the same. The cell becomes less likely to fire, and it serves as a brake that suppresses excitation. Okay. So here we have a more complex reflex arc where it includes both an inhibitory and an excitatory synapse, right? So sensory information comes in, it makes connections with nerves that are going to send excitatory messages to one portion of the muscle and inhibitory signals to another muscle in the leg. So you can just follow the connections here, right? Flexor versus extensor muscles, right? When one's excited and one's inhibited. Now, if there is a severing of this arc from information moving to the cortex, the reflex arc is actually faster and it maintains its integrity. There's feedback coming from the cortex that's going to then moderate the responses of these circuits. Okay, so again, we're looking at this so far as if it's just been a one cell to one cell communication, and neurons make contact with several neurons within networks. And so they can receive different messages. They can receive both temporal and spatial summation, and they can receive inhibitory postsynaptic potentials as well as excitatory postsynaptic potentials. So, so again, it's the cumulative effect of messages coming from multiple neurons within a network. Okay, so how does this chemical translation happen? And it happens with chemical messengers called neurotransmitters. Lowy performed an experiment where he artificially stimulated the heart of a frog by using an electrode. And heart rate increased. He extracted chemicals from the heart. He injected the chemicals into a different heart and measured heart rate, and they matched. And the conclusion was that chemicals are responsible for this part of communication. The excitation or inhibition messages come from chemicals. Neurotransmitters. Neurotransmitters are stored in terminals or vesicles. So terminal buttons, maybe bulbs, maybe vesicles. You'll hear these terms to describe these packets that contain neurotransmitters. And when an action potential reaches these bulbs or buttons, it causes calcium to enter the terminal and the terminal to release the neurotransmitter. So this is called exocytosis, this dumping of neurotransmitters from these vesicles or packets. Now a neurotransmitter is going to float across the synapse, and it's going to bind to receptors on the postsynaptic neuron. And that binding is going to cause either an EPSP or an IPSP in the receiving cell. So I'm going to stop here for now, and we'll continue on in the next video.